All right, so we've gotten through basics about population genetics. You know what an allele is, you know what a polymorphism is, you know what a single nucleotide polymorphism is, you know what a population is. Now we're going to start talking about allelic frequencies. That is, how often those alleles appear in a population. When we're talking about allelic frequencies, you have to understand the idea of a gene pool. A gene pool is the total of all genes, including every possible variant of every allele in a population. Basically, you can imagine it as if some organisms just dropped all of their gametes into a pool and called it good. Probably this is not the best picture. This is not how warthogs mate. They don't just go to a pool and drop their gametes in. It is, though, very similar to how sea urchins mate. Sea urchins just release their gametes randomly into the sea and hope, as much as a sea urchin can hope for anything. I don't know that they have hopes and dreams. I don't know, though. They wear tiny hats, and that's cool. You guys know? That's something I learned. Sea urchins, you know what a sea urchin is? A little spiky ball of death on the bottom of the ocean that you step on and it hurts really bad? Yeah. They, in order to protect themselves and hide, will always put seashells on their heads and rocks, small rocks. They can't stop themselves. I'm very similar. I always put things on my head. Like, I'll end up with a colander on my head for half the day and just forget I have it there. Not like at work. But that's just a home thing. It's, you know, put on pajama pants, put on the colander. Sea urchins are the same way. They'll just stick things randomly on their head. So if you put tiny hats into a sea urchin tank, eventually all of the sea urchins will be wearing tiny hats. Isn't that cool? It does bring me a lot of joy. All right, so quick algebra review. Frequency is expressed as a decimal. We're going to be talking a lot about frequency. You really need to put a 0 in front of that decimal. Don't just put like 0.1. You need to put 0.1. And the reason for that is it's super easy to lose track of where that point is and just put 1. And there's a huge difference between 1 and 0.1. If this was a nursing class, that would be the difference between somebody living and somebody being dead at your hands. So frequency is the number of a specific event divided by the total number of events. Practice problem 1, this is in your lab. If an event occurs 7 out of 10 times, what is the frequency? 70% expressed as a decimal. 0.7, right. So we take 7 divided by 10, and you get 0.7, which is equivalent, you're right, to 70%, but because it's frequency, and we're going to be working with these in mathematical equations, you want to use that decimal. Practice problem 2. All right, so you got the 70%, right? How about this 44%? What would be the frequency? Yeah. 0.44, the end. So far, so good? Questions, concerns? So 44 divided by 100 equals 0.44. All right, now we're getting to a little bit more biology. We have 60 individuals in a population that have one genotype. Out of a population of 200, what is the frequency of that genotype? 0.3. That is correct. 0.3. How did you get 0.3? That is correct. 60 divided by 200 is 0.3. So far, yes? No, you don't. I do not care about significant figures. Yeah, I hate them. So, right, as long as there's a 0 before the decimal point, that's what matters. All right, so we talked about genotype. Let's talk about allelic frequencies. So remember, alleles are the number, the specific form of that gene. So you have the number of alleles of a specific type divided by the total number of alleles present to get your allelic frequency. Remember that most of the organisms that we're going to deal with are diploid. What does it mean to be diploid? You have haploid and diploid, 2, right? Each diploid organism, like you, has 2 chromosomes, so 2 potentials for an allele. One from your mom, one from your dad. This is not, again, not the best picture because it's harder to see, but you've got different mice, I guess. I'm going to say they're mice with little red eyes. These mice all have different possible alleles. They have dark alleles or light alleles. If you have 2 light alleles, you're white. If you got 2 dark alleles, you're black. If you've got a heterozygous genotype, you're also black. So like this one right up here in this corner, this is a dark allele and a light allele leading to a black form. So to get the total number of alleles, we count the total number of organisms and multiply it by 2. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 total organisms. So how many total alleles? 30. All right, we got 30 total alleles. And how many white alleles? Oh, this is going to be a little harder. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So it's 13 divided by 30 will give you your allelic frequency of the white alleles. It's another. Oh, well, there will be another example. We have allelic frequency. We can also look at genotypic frequency. So not just looking at specifically the alleles, but you're looking at the different genotypes. What are the 3 possible genotypes you could have in general?
Homozygous recessive, heterozygous, and then homozygous dominant.
Awesome, Nick. Yes. So homozygous dominant is 1. You have 2 capital letters. You could be heterozygous, a capital letter and a lowercase letter, or you could be fully homozygous recessive, which is like 2 lowercase letters. So you have the number of individuals with a specific genotype divided by the total number of individuals. Now, in Bio 101, we tended to use A's and B's, right? Big A, big B, little b. In this course, we tend to use P and Q. And the reason we use P and Q, we use P for the dominant allele, Q for the recessive allele. And it's because when you look at them, P looks like that, and Q looks like that. It's not like using Z's or O's or T's or S's where they look the same. And it's just by convention. So here we've got organisms, a bunch of, this time they are just fish, yeah. You've got some that are homozygous, or you've got big A. So we got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 big A's. And we've got 1, 2, 3, 4, 5, 6 little A's. Or 6 B's. So we have 14 here. We've got 6 here. What's our total number of alleles? 20. So 14 over 20 equals 0.7. 6 over 20 equals 0.3. Questions? Concerns? Feeling okay on this so far? Math's working out for you? There are mathematical consequences to this. And this is what blew everybody's mind. I kid you not. P, the frequency of the dominant allele, plus Q, the frequency of the recessive allele, must always equal 1. Because you'll have dominant alleles, you'll have recessive alleles in a population. It doesn't matter if you're homozygous, heterozygous, whatever. You'll either be dominant or recessive. You add them together and you get 1, or a frequency of, you'd have 100%, which is 1.0. Questions on where I got that from? Let's go back to this. Well, let's not go back because then I'd have to restart the recording. Think about those fish again, right? We had a grand total of 20. 14 of them were big A. 6 of them were B. 14 plus 6 is 20. So it's 20 over 20, which is equal to 1. P plus Q equals 1. Always remember, P is the frequency of the dominant, Q is the frequency of the recessive. When you're working out these problems, sometimes it helps to have a cheat sheet or something sitting in front of you that has that written down. because it's super easy to lose sight of what these actually mean. So P is the frequency of the dominant, Q is the frequency of the recessive. So, if we know the frequency of the recessive allele in a population is 0.3, we kind of just did this, what must be the frequency of the dominant allele? 0.7. P plus 0.3 equals 1. Move 0.3 to the other side, and you get P equals 0.7. That's problem number 4 in your lab. So yeah, whenever it says practice problem at the top, that's what it means. Here we've got the frequency of a dominant allele in a population is 0.35. So what's the frequency of the recessive allele? 0.6. P plus 0.35 equals 1. P equals 1 minus 0.35. So P equals 0.65. Tie my shoe because I'm going to die. There we go. P plus Q equals 1. So they figured that part out. This was a postulate put forward by a duo, Hardy and Weinberg. It was Godfrey Hardy and Wilhelm Weinberg, which are the most Harry Potter names you will ever hear in your entire life. Godfrey Hardy, not surprisingly, was an English mathematician. Wilhelm Weinberg was a German physicist. They got together at a convention. The story goes, they got together at a convention. They were having a drink at a bar, discussing math, like you do at bars. And they looked at that whole idea of, well, we got P plus Q equals 1. So we know that in a population, a single individual must be P plus Q. The frequent, and these are expressed as, frequencies. Chances of getting P and a chance of getting Q. So what they ended up saying was, if you were to take that P plus Q equals 1, move this down a little. So that is the chances of any one individual, any of you, you'll either have a P or a Q. Put together, you have a 100% chance of having a P or a Q. You mate with somebody that has a P or a Q. 1 times 1 equals what? 1. So they said, from this we can figure out what are the chances of having any particular genotype. You use the FOIL method. Do you guys remember foiling things? I know, it's so long ago. Use the first ones. P times P is P squared. The outer ones, PQ. The inner ones, PQ. And the last ones, Q squared equals 1. And they derived and said, cool, P squared, we got 2 PQs, plus Q squared equals 1. Questions on how they got that? Because now what this does, and it's kind of neat, just show you this. If you've got that, you know P squared. What's P again? The frequency of what? Dominant. So if you have 2 P's, frequency of the dominant and the frequency of the dominant, this is your frequency of the heterozygote. I'm sorry, of the homozygous dominant. It's your chances of getting big P, big P. So frequency of the homozygous dominant. This is the frequency then of what? It's a chance of getting a P and a Q. Your heterozygote. And then what's that last one? Q squared? Frequency of what? Right, frequency of the homozygous recessive. The only time this Hardy-Weinberg works, p squared plus 2pq plus q squared equals 1, is when you have a large population, when you have no migration, so a very, very large population, no migration, so you're not bringing in new alleles, you're not releasing new alleles, letting them go. You have entirely random mating. There are 0 mutations that change your alleles, and there is no natural selection. If those conditions occur, Hardy-Weinberg will always hold true. One generation after the next will always be p squared plus 2pq plus q squared equals 1. Whatever starting alleles you have, the frequencies don't change. Yes? That is basically what it means, yeah. This is what we call a null hypothesis. If this holds true, then evolution is not occurring. And that's going to be the big point. That you totally nailed the takeaway. The takeaway is this doesn't hold true in real life, because in real life, evolution occurs. So over and over and over again, under those very, very specific conditions that, as you've pointed out, don't happen in the real world, this will always hold true. So remember, here's your cheat sheet. P squared plus 2pq plus q squared equals 1, where P is the frequency of the dominant allele. Q is the frequency of the recessive allele. P squared is the frequency of the homozygous dominant. 2pq is the frequency of the heterozygote, and q squared is the frequency of the homozygous recessive. This will start to make more sense as we work through problems. So I'm going to give you some. But before I give you some, like lots of problems, let's ask you an easy one, Sabrina, for funsies. What does p squared represent? Right. So Savannah, is that right? What does p then represent?
Just like regular P.
Just regular p. Yes. So the frequency of the dominant allele. And there's a big difference between the frequency of the homozygous dominant and the frequency of the dominant. The next few problems are going to be asking you to see if you can figure out from having one value what the rest of the values are. So I'm going to start easy. We're going to start talking about butterflies. The color brown in butterflies is dominant, and the color white is recessive. So P is going to be brown. What's the other one? Q, sorry, is going to be white. 36% of all butterflies are white. So if you know 36% are white, what does that tell you right there? 36% are white. What does that represent? Which of those values? P, Q, P squared, 2pq, Q squared. It's not P squared. P squared would be, they'd all be fully brown. Q squared. Right, you'd think, and that's why you have to know the difference between P and Q and P squared and P. We are looking for the recessive genotype, and the only one you can ever tell is recessive, is the homozygous recessive. Having if you knew the number of brown ones, you wouldn't know how many were homozygous dominant versus how many were heterozygous by looking at them. You'd have to grind them up and run some genetic analysis on them. And it's not fun grinding up butterflies. They get dust everywhere. Yeah, it's true. Have you ever touched a butterfly? Well, if you touch a butterfly, you get all dusty. Yeah, that's not fun. Then they die. So don't go touching butterflies. So we got Q squared. What is Q squared equal to now then? What value? 0.36. Good job not saying 36%. So 0.36. If Q squared is 0.36, how do we get Q from that? Oh, now we're getting into real math. That's correct. So to get Q, you're going to take the square root of 0.36, which is equal to 0.6. What can I do with that 0.6 value? What? It's right, convert it to P. How do we convert it to P? Yes, we use numbers. We use one number is going to be 1 minus 0.6, which is going to be equal to 0.4. You're rocking this. 0.4. Boom. So far, so good. Did I lose you guys? This is where I lose people because I get excited about this crap. Okay. Thanks for you're like that's okay. You're good. All right. So we got p. What's p squared? I know somebody's like calculator. Do you know what 4 times 4 is? That's 16. So you're going to move the decimal over because there's 2 points, so it's going to be 0.16. And we got 2pq. 2 times 0.4 times 0.6, which is exactly 0.48. Well done, guys. That's it. That's the whole problem. You solved it. Well, okay, together we solved it. Check this out. If you add p squared, 2pq, and q squared together, what do you get? 1. And we showed it worked. Pretty sweet, huh? Don't worry, there's a lot more. This one wasn't in your lab. I know, this one was just to see if you could do it, and I know you can. Here's another one that I like to do. This is about the ABO blood typing system. You guys know that there's type A blood, type B blood, type O blood, and if you get the wrong blood, you die. Basically, yeah, cool. We do have a blood drive today. You should go give blood. Oh, or not, whatever. In a given population. So let's say in our population, there's nobody with type O blood. There's no even type O alleles. You just have A's and B's, and that's it. I don't know. Some virus came and wiped them out. It was sad. We lost them all. If 200 people have type A blood, 75 have type B, and 25 have type B blood. So 200 people have type A, 75 have type AB, and 25 have type B. We know we'll have, for type A, you have AA, big A, big A. Type B, it's type big B, big B. Let's just count the alleles. How many total A alleles are there? So you have, so say that number again. So we have 200, right, from here. But we also have 200 more from that one. 400, and then how many more? So we have A is 475. B is how many? 125. Cool. So what are the frequencies of P and Q? So we got, what's our total number? 600, right? I'm sorry. Is it 600? Yeah. 200 plus 75 plus 25 times 2 is a total of 600. And 125 over 600. I don't know. What's 125 over 600? It seems like it's like 0.25, but I'm not sure. Numbers. I'm going to say it's 0.25, unless somebody wants to disagree with that. Is it right? Kat, are you checking? Sorry, you had a calculator. I didn't know if you were putting numbers in.
So the second one is 0.208, so 0.21.
125 divided by 600? 0.208 something? We could. So 125 divided by 600. Okay, sure. And then the next one is 0.791. I don't care how you guys round it, honestly. That doesn't matter to me in the slightest. 2 digits. Let's do 2 digits at the most. So there you go, frequencies. You didn't have to go through P squared, 2pq, and q squared. You could just count the alleles. So you got to figure out what type of problem you're dealing with. Oh my freaking goodness. Let's talk about PTC, Phenylthiocarbamide, I think it is. This is a chemical in Brussels sprouts. It makes them taste very bitter to some people. Other people can't taste it at all. So to them, Brussels sprouts taste sweet. So some people, Brussels sprouts are very bitter. They hate Brussels sprouts. Some people enjoy Brussels sprouts. How many of you guys like eating Brussels sprouts? A few of you. Yeah, several of you. How many of you guys really, really hate Brussels sprouts? Right. I'm willing to bet that you can taste this chemical in them. Yeah, there's a biological reason behind you hating it. There you go. Yes, it is. Does it taste like soap to you? Yep, there you go. You can taste this chemical. You sampled 215 people and determined 150 can detect the bitter taste of PTC and 65 could not. So calculate all of the frequencies. P, Q, P squared, 2PQ, and Q squared. All right, so I've given you some time to work on this. We've got 215 individuals. And there it is. 215 individuals total. And 150 can taste it. So 150 of them have what alleles? So it says the ability to taste is due to a dominant gene. So that's going to be having a T. Now it could be big T, or it could be big T, little t. So that value right there doesn't tell us what our recessive alleles are. We need to get to Q squared. So from this, how can we get to Q squared? How can we figure out the number of recessive individuals? I'm sorry, the frequency of the recessives. The frequency of the homozygous recessive in particular. Oh, we have another number, don't we? What's the other number? 65. And we got 65 by subtracting these. So 65 equals 65 over a total of 215 equals Q squared, which is equal to what? 0.3? Great. So how do we get Q? Take the square root. Q equals the square root of 0.3. If Q is the square root of 0.3, what's P? Don't even worry about doing the math on a calculator. Just based on what you know from the formulas, what does P have to be? Sort of 1 minus the square root of 0.3. Yes? Oh. So if you took 150 over 215, if you were to do it that way, you would end up actually getting P squared plus 2PQ. Because you can't tell the difference. That 150 includes all of the homozygous dominant and all of the heterozygous. And now you got two variables and you can't solve for it well. So you want to always, always, always get back to Q squared. If you have these two, P squared plus 2PQ, you could take this, so you could do 1 minus that, and that will give you Q squared. Because basically, you've got that big P squared plus 2PQ plus Q squared equals 1 equation, right? So if we're trying to figure out Q squared, all we need to do is move these two to the other side. So now we've got Q squared can equal 1 minus P squared. Well, we'll just put minus 2PQ, which is what that value is that you solved. So you got 150 over 215 is P squared minus 2PQ. Well, there, we'll do it that way. Put it in parentheses. Makes it a little bit easier to understand. So if you subtract this value from 1, you get Q squared, which is the same way of getting 65 over 215. Make sense? Mostly. Sort of. No, not at all. We can work through another one. So we got P, we got Q. We can figure out from here P squared 2PQ just by substituting in. Let's work on a slightly easier one. I will tell you, I think this is probably the hardest it's going to get. This one. Albino squirrels. Albino squirrels will mess you up. Albinism is a recessive trait. If a population has 4% rate of albinism, so 4% of all of them are albino, they are homozygous recessive, what would be the expected genotypic and phenotypic ratios of the remaining population? So figure out p squared, 2pq, q squared, q and p from that. Give this a shot. It's a little bit. It's a lot easier than that last problem. All right, so let's take a look at this. The first step in this: if the population has an albinism rate of 4%, so that means that is the albinism is a homozygous recessive trait. So that means that what would that 4% represent? Which of these variables? Yes. Yeah, exactly. Yes. Q squared. So Q squared is 0. And this is where a lot of people make a mistake. It's 0.04, not 0.4. Okay, good. As long as you're there. All right. So if we got Q squared, what can we calculate next? Q. How do you calculate Q from Q squared? You'd square root. Square root of 0.04 equals 0.2. All right, now we've got Q, what can we calculate? P. How do you calculate P from Q? Right, so 1 minus 0.2 equals 0.8. And now we're just plugging in. So P squared, 0.8 times 0.8 is 0.64. And then 2 times P times Q, so 0.16 times 2 is 0.32. You get P squared from, you got P, so you make P times P, so 0.8 times 0.8 is 0.64. Did you get 2pq? You could get 2pq by solving for this and then subtracting them from 1. Yeah. And you can do that because it's Hardy-Weinberg. It's all of these things equal 1. In a perfect world that doesn't exist. Questions or concerns? 2pq is 2 times 0.8 times 0.2. So 0.2 times 0.8 is 0.16 times 2, which is 0.32. And that's just math. That, you can use a calculator for that. You will always have a calculator on these tests. So there you go. You can use your phone. I don't care. All right. Let's try another one. This is one that's in the lab. After sampling a few hundred individuals in a large population of butterflies, a scientist calculates the frequency of a recessive allele for yellow wings as 0.3. So you have the frequency of the recessive allele. What's that equal to? If they tell you the recessive allele, Q, not Q squared in this sense. So you got Q. So they want to know what's the frequency of the dominant allele. You guys, come on, off the top of your head. 0.7, right. Sorry, I was just like, yeah, you got this. Assuming the population's in Hardy-Weinberg equilibrium, you can calculate the rest of the genotypic frequencies. So we've got P equals 0. We start with Q equals 0.3. Thus, P must equal 0.7, 1 minus Q. P squared has to equal 0.49, 0.7 times 0.7. 2PQ has to equal 2 times 0.7, which is 0.14, times 3, which is, 0.42? 7 times 3, 21 times, 0.42. And then Q squared is 0.3 times 0.3, which is 0.09. Add them all together and they equal 1. Yep, add them all together and they equal 1. So this actually just goes through step by step how to do it. All right. So we've now stumbled upon one of my my biggest fears, which is the brain-eating amoeba. It is wildly it is unlikely that you will catch a brain-eating amoeba in your nose. Basically, in order to get a brain-eating amoeba to eat your brain, you have to be in warm water, and that warm water has to go up your nose, and then the amoeba will cling onto your olfactory nerve, crawl along it, get into your brain, and start to digest your brain as it rapidly multiplies in your brain. And I believe there's a 99% fatality rate. So you get it, basically you die. Yeah. Yep, my mom is always getting on my case. She's like, "You should use a neti pot for your allergies." And I was like, "Do you understand how brain-eating amoebas work?" Just the other day, I'm still counting down days because it could take up to 2 weeks. I was in the shower and I leaned my head back and hot water went up my nose and I was like, "No, brain-eating amoebas!" I know, because they're basically everywhere. If you go find a puddle of water, there are brain-eating amoebas in that puddle of water. So don't stick your face in a bottle of water and take a deep breath. Right? Usually they hang out in the warmer the water, the more there are, and the more sediment there is, the more amoebas there are. So now you know some of my greatest fears. And we decided to make an entire problem about it so I get to face it every semester. A scientist is studying a set of brain-eating amoeba in a pond and has discovered there's a set of alleles. The dominant form makes it target fish. The recessive form makes it target humans. The scientist tests 200 amoebas and finds that 10 of them are the human brain-eating form. So we have a total population of 200 amoebas, of which 10 are brain-eating. So what does that value represent? Yes? That is Q squared. Well done. So now we got Q squared. This is our frequency of the recessive genotype. It's also the recessive phenotype because genotype and phenotype are the same when you're looking at the recessive. All right, so if we have Q squared, how do we get Q? So Q equals then the square root of 10 over 200. Assume to estimate the allelic frequencies. So all we're looking for here are the allelic frequencies. We are not looking for P squared, 2PQ, and so on. P then is going to equal 1 minus the square root of 10 over 200. You could. I don't care. The lab, I think, probably puts it in a decimal form. So you'd have to do the math on your calculator. In fact, let me go ahead and erase this because, check this out. It goes step by step through. It lets us calculate Q squared. So Q squared is 10 over 200, which is 0.05. Q is then the square root of 0.05, so Q equals 0.224. I know you guys looked at me like, oh, he erased it, now I'm doomed. No, I just gave you the decimals. Because P plus Q equals 1, if Q is 0.224, P plus 0.224 equals 1, so P has to equal 0.776. You surviving? Questions? All right, so this time science is studying a population of butterfly fish. It's commonly observed on coral reefs. They discovered a set of alleles. The dominant form gives the fish a white color. The recessive allele causes them to be green. She samples the reef population and observes the following. Out of 2,500, 225 are green. Estimate the allelic frequencies for P and Q. You guys work on that. All right, so to figure this out, again, we ask, what do we know? We know the number of recessive phenotypes and the total number of organisms. That lets us calculate Q squared. So we know Q squared equals 225 over 2,500, which is equal to 0.09. I know, you're rocking this. So Q is the square root of 0.09, which is 0.3. P plus Q equals 1. P plus 0.3 equals 1, so P equals 0.7. Yeah. That's all you needed to find out for this. 0.3, 0.7. Moving on. 5 years later, she comes back. She's looking at the exact same reef, but this time she counts 2,000 fish. 720 are green. So we've got different numbers this time. Estimate the allelic frequencies for the population 5 years later. Give that a shot. All right, so this is 5 years later. What do we know? We know the number of recessive phenotypes and the total number of organisms. So we could take, to get Q squared, we're going to take 720 divided by 2,000, and we get 0.36. We take the square root of 0.36 to get Q, so we have Q equals 0.6. If Q is 0.6, what must P be? 0.4. So the trick to this one, there's no trick to this one. This is the exact same problem as the last time with different numbers. Say that again? We're good. We're good with that so far. Now here's the deal. This question asks you, has evolution occurred? And this will be the first time we note what evolution is and why all the time people are like, they want to debate me on if evolution occurs, which is kind of weird to debate me on. I will tell you guys, I don't talk about evolution at all in Bio 101 because I don't feel like the Bio 101 students have the mathematical background to understand what evolution is. But you guys do now. Evolution is occurring if allelic frequencies change. The definition of evolution is a change in allelic frequencies from one generation to the next. So in this case, has evolution occurred? Look at your first problem. What were the allelic frequencies? 0.7, 0.3. What are they now? 0.6, 0.4. Are they different? Yes. Thus evolution. The end. There is no arguing with it. It happens. Like, what if it didn't? We would be in perfect Hardy-Weinberg equilibrium. So you can actually show, is evolution not occurring or is evolution occurring? It is rare, but you can find conditions in Hardy-Weinberg equilibrium. Usually those are completely controlled. It is in a zoo, and you only have so many alleles, and you don't let other alleles in. Likely not. Hardy-Weinberg counts as what we call a null hypothesis. We disprove Hardy-Weinberg in order to show something else is happening. This is a fun one. I'm going to walk through this one with you because it's a little bit confusing. So after graduation, you and 49 of your closest friends, as if you had 49 friends. So 25 males, 25 females, charter a plane to go around the world. You all crash and land safely on a deserted island. No one finds you, and you decide you're going to start your own new civilization because to hell with the rest of the world. Unfortunately, 9 of your friends are carrying, that is, they are heterozygotes for the sickle cell anemia trait. They don't have sickle cell anemia, they're just fine, but they have one allele that's kind of a little bit broken. Assuming that the frequency of the allele does not change as the population grows, what will be the incidence of sickle cell anemia on your island? That is, how many will be Q squared? So in this case, we've got a grand total of 50 people. So if there are 50 people, how many alleles are there? 100 alleles. We know 9 of those people are carriers. That means that they are basically big A, little a. If 9 of them are big A, little a, how many recessive alleles are there? 9, right? So you have 9 people that look like this. So big A, little 1, 2, 3, 1, 2, 3, 1, 2, 3. And you just count them. 1, 2, 3, 4, 5, 6, 7, 8, 9 of them out of 100 have sickle cell anemia, which is Q squared. So if 0.09 equals Q squared, what must Q equal? 0.3. That's just taking the square root of 0.09. Here, I'll write that step just so we can be clear. That doesn't make it much more clear. Okay, so if Q equals 0.3, must P equal 0.7. Now it asks, what will be the incidence of sickle cell anemia on your island? Oh, guys, I messed up. No, did I mess up?
Isn't it for the heterozygous?
Yeah, wouldn't it be 2PQ?
It would be 2PQ, yeah. So, but that's not what I'm looking for. What I'm looking for is just Q squared off of that. So I went way too far. All I needed was this value. Basically, you'd end up with what will be the instance of sickle cell anemia? 9 out of 100 people. So that is equal to, oh, that's not Q squared. See, I told you this was a rough one. That's actually equal to Q. So Q squared equals that, whatever that value is squared. Sorry for screwing it up, guys. I'll definitely make sure to delete this slide. Let me walk through it again, okay? Now that I screwed you all up by putting the wrong values. So we know that there are 9 out of 100, which gives us Q. All we want to know is Q squared. What is the incidence of sickle cell anemia? Which is equal to 9 divided by 100 squared. Anybody have a calculator to calculate that? Thanks, Kat.
It's 0.0081.
So your incidence of sickle cell anemia is going to be about 1% of the population. Yes?
So, I was gonna say like I thought when we squared Q earlier it was 0.3, but not a B.
Yeah, it's because I screwed up last time. Yeah. It was my fault. I'm sorry for messing that up. Why don't we do another part of the homework problem set? Call it done, right? Except it's not going to be done. There's still so much more. What's the estimated allelic frequency of the dominant allele in the population if the frequency of the homozygous recessive genotype is 0.25? Back to what we were just doing, right? We ask ourselves, what do we know? The number of the recessive phenotypes and the total number of the organisms. So we're going to calculate the frequency of Q squared. Well, we don't need to calculate the frequency of the Q squared because it is Q squared equals 0.25. Because this says the frequency of the homozygous recessive genotype. That is Q squared. If that's Q squared, what's Q? Square root of 0.25. What's that, Kat? You're working it out?
Sorry.
You,
That's a half.
it's a half?
0.5.
0.5. Thank you. All right. Oh, this is going to be easy, right? If Q is 0.5, what's P? 0.5, because 1 minus 0.5, 0.5. So what's the estimated frequency of the dominant allele? It is 0.5. You guys surviving this? All right, what's the estimated frequency of the heterozygous genotype in a population if the frequency of the recessive allele is 0.36? So we want to know the frequency of the heterozygous. Heterozygous is 2 times P times Q. We're given the recessive phenotype again. That's awesome. Always try and get that recessive phenotype because that will allow us to determine Q squared. So Q squared is 0.36. Frequency of the, I'm sorry. The frequency of the recessive allele is 0.36. So Q is 0.36. If Q is 0.36, what's P? 0.64. So we got P, we got Q, so we got 2 times 0.64 times 0.36, so we get 0.46. All right, what are the estimated allelic frequencies in a population if the frequency of the dominant phenotype is 0.64? So we have the dominant phenotype this time. We do not have the recessive phenotype. We need to get the recessive phenotype. How do we get the recessive phenotype? What? Numbers. Numbers are the way to get that. Yes.
So they're giving us PQ.
They're giving us 2pq and p squared. So remember that last time how you solved it? You're solving it the exact same way.
Yeah. What?
Whoa, whoa, whoa. This is crazy talk. We are given the frequency of the dominant phenotype. That is, we're given all of them look, I don't know, brown. How do we figure out then what percentage are white? That recessive.
1 minus 0.64.
Yes, that's it. It's just one more step. You take 1 minus 0.64 and you get some other number, 0.36.
Wait, so what is it they're giving us at first?
They are giving you the dominant phenotype. So it's not just p, it's p squared plus 2pq is what they're giving you to begin with. So that's equal to 0.64. That in itself doesn't tell you anything because you're dealing with two variables. You need to get q squared. Right. So if we looked at that whole Hardy-Weinberg equilibrium problem, we know that q squared is equal to 0.64. Well, it's 1 minus these two, which is equal to 0.64, which is equal to 0.36. Oops, and that's q squared. And if q squared is 0.36, well, it must be q. Q is 0.6. So far, so good? I lose anybody? If Q is 0.6, what must P be? So P must be 0.4. This question is asking what again? What are the estimated allelic frequencies? Boom. P and Q. Yes?
How do we get 0.6?
How do we get 0.6? We took the square root of 0.36. I'm going to erase it off the board and show you typed up and pretty on the slide. So we look for, we know the dominant phenotypes, we know the total number of organisms, so we need to calculate the frequency of the recessive because we're always looking for that recessive, which is 1 minus the dominant phenotype, which gives us our recessive phenotype. That's equal to q squared. So q squared equals 0.36, thus q must be the square root of 0.36. So q equals 0.6. P plus q equals 1. P plus 0.6 equals 1, so P must equal 0.4. And then from there, you can calculate the genotypic ratios if you really wanted to. This problem doesn't ask for that. We're getting there, guys. We're getting there. A population of centipedes. Centipedes are poisonous. They are predators. They will take down worms and they will bite you and it will sting. They find that a recessive allele that, when expressed, makes the centipede's saliva venomous. Do you guys know the difference between being poisonous and being venomous? Yes.
If you eat it and it makes you sick, it is poisonous. If it bites you and makes you sick, it is venomous.
That's exactly right. So if it bites you and you die, it was venomous. If you bite it and you die, it was poisonous. So from here, we need to know, what do we have? We know the number of recessive phenotypes, and we know the total number. So we found the recessive allele is 0.4. If the allele is 0.4, the recessive allele is 0.4, what must be the dominant allelic frequency? 0.6. So we know Q is 0.4, thus P must be 0.6. Wow, I should not have done that in yellow. Yeah, sorry about that. I can't even read it on this screen. So what would be you expect the frequency of the venomous centipedes to be? So we know that Q is 0.4, so Q squared equals 0.4 squared, which is 0.16. In this case, we were looking for the frequency of the venomous centipede. So we were looking for Q squared. We were given Q. So Q times Q is Q squared, which is 0.16. Questions on that? I think there's another onto this. Yeah, same population of centipedes. What would you expect the frequency of the heterozygous genotype to be? Well, in the last one, we figured out P and Q. So we had Q at 0.4, P at 0.6. We wanted to know the frequency of the heterozygous, so it's P times Q times 2. 2PQ is 2 times 0.6 times 0.4, which is 0.48. We're just building on what we did in the last problem. We calculated this in the last, well, we were given this. We calculated P, and then we just put them all together. Step by step, we're getting there. Questions? Yes?
For the first time, are we only going to have to answer for like 2PQ, or do we have to put in all the equations?
I think it's just going to ask you for the specific. Some of them will ask for all three of them, but those will be the ones that say give all of the values. Give the genotypic values, yeah. All right, back to more centipedes. They head out to the field. They find out that this person's just obsessed with venomous centipedes. They find out the actual frequency of the venomous centipedes does not match Hardy-Weinberg equilibrium. What does that mean is happening? You win! That's it. Evolution must be occurring. Whenever allelic frequencies differ from what you expect in Hardy-Weinberg, evolution is occurring. Applying it to a real-world situation. Sickle cell anemia is a genetic disorder caused by a recessive allele. Basically, it's a SNP. It's a single nucleotide polymorphism. There is one change in the billions of nucleotides in your genome. That one change means the entire shape of the blood cell has changed, and it's no longer able to easily carry oxygen, and it's got this weird shape to it that causes it to clog up in your arteries, leads to it's fairly painful. Yes, it looks like a banana. Thank you. Out of 100,000 people, 1,440 have the disease. So we have 100,000 people and 1,440 have it. What does this value represent? Q squared. So in this case, even though it's a big number, or actually a small number, how do you get Q? You take the square root of it. And because I apparently am absolutely stuck on relying on you, Kat, you know what that value is? 0.12? I can totally rely on you. That's great. All right. So this is now equal to Q. We're trying to figure out how many are carriers. That is, how many are heterozygous. Can we get it yet? No. What else do we have to calculate? When we got Q, we may as well calculate it. P. So if Q is 0.12, what must P be? 0.88. So how do we get the frequency of the heterozygous from that? What do we have to do? Say it again? Not squared. No. Yes. 2 times 0.88 times 0.12. So we've calculated it out. We figured out what percentage actually have the, what frequency have the disease. We calculated out Q squared, which gave us 0.0144. We took the square root of that, and we got Q is 0.12. That meant that P is 0.88. So to finish calculating it all out, 2 times P times Q is 2 times 0.88 times 0.12, which is equal to 0.2112. That is our frequency of heterozygous. Is that what the problem is asking for? No, it's not. What is it asking? How many people in that population of 100,000 are carriers? Well, we have a frequency of 2.2112. How do we figure out how many of those 100,000 are carriers? Right, so you multiply 0.2112 by 100,000, which gives you 21,120 carriers. It's a lot of carriers for a very small number of infected individuals, or afflicted individuals. Questions? Oh, man, so many problems. For goldfish, solid orange coloring is dominant, while orange with black spots is recessive. You have 2,500 total goldfish stocked in an outdoor pond. Of them, 2,275 are solid orange and 225 are orange with black spots. Which one of those, again, is the recessive? The ones with black spots. They came from a protected indoor stable environment, so it's in Hardy-Weinberg equilibrium. Determine the allelic and genotypic frequencies of that population. I'm going to let you guys work on that. We know from this problem the number of recessive phenotypes. We also know the number of dominant phenotypes and the total number of organisms. Never, ever, ever care about dominant phenotypes. The only reason you want to use a dominant phenotype is to figure out what your recessive phenotype is. The recessive phenotype is the only one you can determine by looking at it. Otherwise, you're not just looking at p squared, you're looking at p squared plus 2pq, and that's too many variables to look at. So let's just worry about recessives, q squared. So we know 225 out of 2,500 are orange with black. That gives us q squared, so it's 0.09 is q squared. If q squared is 0.09, q must be the square root of 0.09, which is equal to 0.3. We take q to figure out p. P plus q equals 1, so p must equal 0.7. And now that we've got p and q, we can calculate everything else out. P squared 0.7 times 0.7 is 0.49. So that's the frequency of the dominant genotype, the homozygous dominant phenotype. 2pq is the frequency of the heterozygote, which is 0.42. So 2 times 0.7 times 0.3. And of course, we knew q squared is 0.09. So that's all of it, the allelic and genotypic frequencies. Okay? Ten years later, time passes. The pond was drained. All the fish died. 2,000 fish were recovered. 1,280 were solid orange, and 720 were orange with black spots. We're going to assume that the population settled into a new equilibrium. So what are the allelic and the genotypic frequencies now? All right, again, we're given two distinct, we've got a total number, but also two distinct other numbers. Which of these two numbers do we care about? The 720. So we're going to take that 720 and divide it by 2,000. So 720 divided by 2,000 is 0.36. That is our q squared value. Anybody have questions on how we got to that value? If that's the case, q has to equal the square root of 0.36, which is 0.6. So if q is 0.6, what must p be? 0.4. So we got p, we got q. P plus q equals 1. Q is 0.6, so p must be 0.4. And then we just plug everything in. P times P is 0.16, so P squared is 0.16. 2PQ is 0.48, and Q squared, again, is 0.36, as we saw earlier. Questions on how we did that?
We're getting a lot of the same numbers for each one.
Yes, we are, because there's only so many perfect squares below 100. And it makes it easier for you to do the calculations without having a calculator. So you'll see a lot of 0.25s, a lot of 0.36s, 0.64s, and 0.81s. All right, so here's the big question. Over that 10-year period, has evolution occurred? Okay, fine. You guys are, I'm done. Year 0, you have different allelic frequencies than the year 10. The allelic frequencies change, and thus evolution has occurred. You guys are like, boom, mic drop. That was it. Questions?